Next speaker is Dr. Roxana Myers. Her topic is management of plant parasitic nematodes in ornamental crops. Roxana Myers received a BS degree in tropical horticulture from the University of Hawaii at Hilo and a PhD in plant pathology, specializing in nematology, from the University of Hawaii at Manoa College of Tropical Agriculture. She is currently employed as a research nematologist at the USDA Daniel K. Inouye U.S. Pacific Basin Agricultural Research Center, where her responsibilities focus on the development of cost-effective and sustainable approaches to the control of plant parasitic nematodes. Her areas of research include evaluating methods of media disinfestation, testing the efficacy of nematicides in Hawaii's agricultural systems, utilizing predatory nematodes and entomopathogenic, you can say that when you come up here, nematodes as biological control agents, and developing crop plants with resistance or tolerance to plant parasitic nematodes. Thank you, Dr. Myers.
Hi everyone, thank you for that introduction, and thank you so much, HFNA, for inviting me here to talk about my research. And I look forward to this meeting every year, and I have to tell you, this keeps me on task because I know 1 year from now I'm going to have to report again on my research, so I have to get to it. Today I'm going to give you an update of some of the projects we've done relating to ornamentals. First, I'm going to start off just with the introduction. For anyone who hasn't heard me speak before, I have to tell you what a nematode is, because when I go out there in the community, if people don't have that as a problem in their farm, they have no idea what I'm talking about. So there's a lot of different kind of nematodes out there. They're mostly microscopic roundworms that pretty much fill up every niche of our planet. But I'm going to be talking mainly on plant parasitic nematodes, even though, as Sharon mentioned, entomopathogenic nematodes are also an interest of me, and those are insect parasitic nematodes. But I'll save that for another time. So today we're just going to focus on these nematodes that are persistent in the agricultural production systems and that cause problems. But anyway, you can see from that nematode right there, at its mouth part is a very distinct stylet. And these nematodes use this stylet to puncture the plant cells, and then that's how they uptake their nutrients. So you can tell what type of nematode it is by looking at their mouth part. And they cause billions of dollars of damage worldwide in agriculture. As unknown as they really are troublemakers. So how do nematodes damage crops? Because they're so tiny. Well, there's thousands of them in the soil eating the roots of your plants, so they destroy the root system, and this causes problems with the uptake of water and nutrients, causes stunting of plants and poor crop yields. And because of these symptoms, similar to drought or also nutrient deficiencies, a lot of times they go misdiagnosed. In anthuriums, they reduce the number of leaves, which subsequently reduces the number of flowers, and also the size of the flowers. And this is a picture right here of an anthurium root that's infested. So if you look on the root and you see the little brown necrotic spots, that's the beginning of a nematode infestation. That's where the nematode goes inside. And the nematode will feed on the root and cause destruction, and eventually you'll get root rotting, and saprophytic fungi can enter where the nematode entered, and so you see a lot of black root rot in heavily infested anthurium fields. Okay, so this is the nematode I'm going to talk about today. My favorite nematode, I think it's the most beautiful of all. So I have this nice video, so you guys can hopefully feel the same way. The species is Rhodophyllus similis. It's a migratory endoparasite, and what that means is this nematode goes inside of the roots of the plants, and it just moves throughout the root, feeding and laying eggs throughout the root system. And what's even more strange in anthurium is that these nematodes like to go inside of the gobel, but that causes a lot of problems for us if we're using the shoot cuttings, using the gobel as propagative material, because sometimes even symptomless cuttings can have nematodes inside. And if you take that to a clean field, you're going to start a nematode infestation. So if you can, don't get them. Start clean and stay clean if at all possible. You want to start with clean volcanic cinder. If you can bring in cinder that has been steam sterilized, that's a great idea. You want to watch water movement from the surrounding beds and fields. And everybody knows with all the rain that we've been getting lately, the water has really been coming down, and nematodes love to surf, and that's like their primary method of getting around, besides bringing in infested plant material. And you want to train your staff in good sanitation practices, because I'm sure most of your workers have no idea what nematodes are, and they're not even thinking about these microscopic worms that are down there in the soil. If you can, start out with clean plant material, use tissue culture plants. If not, there's a disinfestation method where you can clean your gobo of nematodes using a hot water bath for 122 degrees Fahrenheit for 15 minutes. Will kill any nematodes, and if you extend that to 24 degrees, send that to 24 minutes, that'll also kill any blight infection that may be in the cuttings as well. And recently I've been really excited because one of our cooperator nurseries started to incorporate this technique into their farm with really impressive results. So I would encourage anyone that can't get tissue culture plants to look into this. But I have to say one warning, and this is a big problem. Once you clean your material, once you heat it, you have to cool it off immediately, because if you don't put it in cold water right after, it's going to just continue to bake, and then you no longer have viable plant material. So there's a fine line between killing your plant and killing the nematode. Okay, so as you heard, we have a lot of projects going on with Anthurium, but some of them are more long-term than others. So I'm just today going to discuss with you some field trials that we've been working on using a new nematicide with the active ingredient fluopyrium. And this project was initiated by Brian Bush and Joanne Lichty at the University of Hawaii CTAR's Komohana Extension Service. And they asked Kathy and I to join in and help with the nematode evaluations. So they've done a lot of work in this project, and we're also cooperating with Dr. Arnold Hara and Dr. Brent Sipes. So this is definitely a group effort, as a lot of people have said today. All of our research projects are. It takes more than one. So these are the new nematicides that are out on the market. This product, fluopyrium, was first released as a fungicide in 2004. And then in 2007, and then in 2014, they realized that, this actually works on nematodes too. So it was introduced as a nematicide. The two products that we're looking at are Indemnify, which is currently registered for use on turf grass, and Luna Sensation. And what's interesting about Luna Sensation is it also has an additional fungicide. And because I see a lot of black roots, rotting roots in Anthurium fields, I thought this might be, very helpful. So this is the first trial, and all of our trials were conducted on a commercial grower farms with our cooperators. And this field was mature starlight, which had already been in the ground for 2 years and was stunted from a bad nematode infestation. We worked with 52 plants per treatment and 4 reps. The application was Indemnify at 0.39 floral ounces per 1,000 square feet. And this was applied every 3 months. And we did a pre-application nematode sampling, and after that, every month we would sample the cinder directly around the root system, as well as taking off some of the roots themselves, and taking them to the lab and trying to do some nematode counts and seeing if we could see any differences with that. Luckily for us, the growers are so helpful that they actually did the weekly harvesting of the flowers for us and did the sizing. And I have to tell you, that's not an easy thing when you're just supposed to be out there harvesting, and every few plants, you have to stop and write it in a little book and count them. So we're so grateful to have these wonderful cooperators who are willing to help us with this research. So this is what the nematode numbers look like. When we took the nematodes from cinder, we weren't able to see any significant differences. And the reason for that is because this nematode, as I mentioned, is a migratory endoparasite. So you're mostly going to see it inside of the root, not outside, in the media itself. This is the results, just an average each month over a 14-month period. And as you can see, with the Indemnify treatment, overall, there was a significant difference between the amount of nematodes was reduced compared to the non-treated controls. But nematode populations are a very difficult thing to use when you're trying to estimate the efficacy sometimes of a treatment. So if you look at the top blue line is the untreated control, and the bottom is the treated indemnify. And as you can see on month 4, that's after 2 applications, the nematode numbers dropped significantly. We had a 53% reduction in the nematode populations at that time. However, as the nematodes went away, the plant started growing more, the roots started growing much more, and that means you have a lot more nematode feeding sites, so you can feed more nematodes. And in the meantime, the controls are getting less and less roots because all these nematodes keep eating them, so you have less nematode populations because they can't survive. So they were all moving to the indemnify side. So as you can see, over time, it kind of leveled out, and we didn't see any differences with those. But what was the most exciting, and I just loved this trial right here, was that the flower size and the flower yield. We really saw a difference, and that's where it counts, and that's what the growers want to see. So we had a 53% increase in the number of flowers harvested on average over the 14-month period. And the most beautiful part of it all was if you look at the number of larges, we had 2 in the untreated control and 36 in the treated, and that's a 1,700% increase in the number of large flowers. So it's like, is that right? That sounds like such a large number, but you only had 2 in the other one. So large flowers, of course, are better, and everybody wants that. And even with the medium-sized flowers, there was, quite a bit of difference, 89 compared to 219, mediums harvested. So flower size was significantly affected, and that's what, was really hurting the nematodes. So this idea that you're getting a 50% reduction in your yields when you have nematodes in your field and you're not treating them, is really important to know. And those numbers are the same as there was a paper by Dr. Aragaki back in 1984, and he said they were causing a 50% yield. So sometimes you don't really notice that because it happens slowly over time. It's not like a bacterial blight infection where you go out one day and you see all your plants going down. This is a slow decline disease. So I just wanted to show, this is the average weekly flower harvest over time. And so you can see around week 24, which is after that second application, I think it's really important you need these subsequent applications, that all of a sudden the indemnify, which is the red one on the top, started to increase in the yield. So it took a little while, 6 months for us to see a difference. But over time, look, the red is getting taller and taller. So if we would have kept on with that trial, we probably would have seen, a lot more results over time. Oh, and this is a really cool app if nobody has used this, especially for researchers, because it's so hard to quantify the growth of your plant. And this is called Cannopale, because we can't be going out there measuring every plant or counting every leaf, when these kind of big field trials like this. And what's great about this is you can use it with your phone, and you just have to go out there and take a photo on a horizontal plane at the same height every time, and then you put it into this application, and it'll tell you the percentage of green canopy cover. And this was great for us that we could use this because we could see when we went out there that the roots were just on top of the surface, the roots were much healthier, but how can we quantify that to show you those results? And so this was the result of the numbers. When we put them into the machine, we had a 43% increase in the amount of leaf mass. So that turns into plant vigor, and so your plant is healthier. And if your plant is healthier, it's also more tolerant to nematodes, and you're going to get less damage. So that was a very, very exciting trial, and I'm so happy I got to share that with you this year. So we're going to go on to talk about our second field trial. So we decided after, we were already seeing good results at the beginning, okay, let's expand this a little more. Let's try out this new product, Luna Sensation, as well as the Indemnify. So we did a little bit bigger trial. We looked at a mature Midori planting. And we did the nematode sampling this time quarterly of the cinder and the roots. And weekly flower harvest and sizing by the growers. Thank you very much for that. Okay, so here's what the nematode numbers look like. Let me first start out by saying there was 2 different nematodes, so that threw in a little bit of twist. So the light green on the top is the root knot nematode, which is also, you do find that occasionally in anthurium plantings. And as you can see, the light green is a little bit bigger in the untreated control than the other two. We didn't see any big differences in the nematode numbers, and I'm going to show you why in the next slide with Indemnify, but the Luna sensation had quite an effect on the nematode populations. Okay, so the green is the Luna sensation, and you can see after one application, the numbers dropped and then stayed low throughout the entire time. However, with Indemnify, it is the exact same thing that we saw in the first trial. After 2 applications, the nematode numbers dropped, and as the plants got healthier, the nematode numbers went up again. So I wasn't too surprised with that. I know this looks super exciting with these nematode deaths here, but I have to say, there was an error with the label rate. So this Luna sensation, unfortunately, was applied at 2.5 times the label rate. So that's probably why it looks extra good. But I'm sure that would have some, positive effects even at the proper label rate next time. So for this one, we only let this run 12 months, and then we wanted to start another project where we make sure everything is how you guys will be using it. But as a proof of concept, wow, I was very excited with these nematode numbers, because I've worked on this for 9 years, and this is the first product besides steam sterilization at 212 degrees Fahrenheit that actually, killed some nematodes. So I've been really excited all year about these field trials. Okay, so here's the flower yields. We didn't see a lot of differences in the overall yields, unfortunately, and there's a few reasons for that. First of all, this is Midori. It's a little bit more tolerant compared to other cultivars, especially the Starlight. So you're not going to have as much nematode damage in these Midori cultivars. In addition to that, the plants were healthier to start with. If your plants are bigger and healthier, they can sustain the damage better from the nematodes. So that's always helpful. If you keep your plants at optimum condition, you'll have less yield reductions. And thirdly, this farm was located in the area of Kapoho near the current eruption site. So we had a lot of inconsistencies in the data taking, because I know there would be days when they were out there and sulfur dioxide would come in and everyone would have to leave work because it was unhealthy worker conditions at that time. So I don't put a whole lot of emphasis on these yield data, so don't feel too discouraged because the numbers weren't as great as the first trial. Because if you look at the extra larges, there were significant differences there. We see you only have 17 in the untreated versus 42, and in the Indemnify, which is quite a bit more, and even 70 in the Luna sensation. So we're getting the size differences, and then that's really important too. Okay, so here's the Cannopale app, and then you can see just by looking at the leaf coverage, the treated ones have more plant vigor. So look at the Luna, you're getting an 89% green canopy coverage compared to 60% in the control and a 75% with the Indemnify. So overall, we've seen the same kind of thing. The plants were, bigger, they were healthier, less nematodes, and the flower size was bigger and the flower yield was better. So we're really excited about this new fluopyram, and it looks like it does have a lot of potential to mitigate some of the damage that's caused by these burrowing nematodes in anthurium. So because we're so excited about it, we're going to keep going. So we're doing an even bigger trial this time, which we've already started at a different grower cooperators, and it's with the cultivar NPR. And we're going to be we're doing it a little bit different. We're going to be looking at Indemnify, as well as this product called Melicon, which is Pacilliomyces lilacinum, which is a biological, it's a biological product. It's organic, it's labeled organic, and it's a fungus. And so this fungus can control the juvenile stages of the nematode, as well as it parasitizes eggs, which, the fluopyram, can't get to the eggs. It doesn't affect them. So we're trying that separately, as well as in combination, and with 2 different rates. One would just Indemnify 1 time a year at the maximum rate, with this application every 3 months of Melicon, and the other by rotating Melicon and Indemnify every 3 months. So we're going to be sampling the cinder and the roots quarterly, as well as looking at the flower yields again. So quite a similar project, but we want to make sure, different cultivars, different locations. We want to try to get this frequency right and get these rates down. And then in addition to that, we're going to be doing a nematicide box trial. And see, I like to do these box trials because I get to add nematodes. See, the growers never let me go onto their farm and put my nematodes out, and I just don't know why, but this way I can kind of control the levels, so I can put higher populations, and I really want to see those nematode numbers reflected, when I do these applications. So this gives me a little more control. There's another new nematicide product out there called Nimitz. The active ingredient is fluosulfone. We'll be looking at that. As well as there's a product called Azosol, which is a neem product, 6% azadirachtin. And I had some interesting results with tomato and root knot nematode with this product, so I wanted to add that in as well. And we'll look at the Indemnify and the Melicon in this trial. So see, I get to put 2,000 nematodes per plant to start. And I think that's all I want to thank. Of course, our grower collaborators, Floral Resources, Pacific Floral Exchange, and Greenpoint Nurseries, because without them we couldn't put it on these big trials, and all their assistance has been really helpful. And all of our UH collaborators and people at the lab, and especially in my unit, the research leader, Dr. Tracy Matsumoto, and my colleagues, Dr. John Suzuki and Lisa Keith. So I have to show you one last nematode video, and then if there's any questions, I'll take those now. Yes?
I used to hear beneficial nematodes to combat this damage-making nematodes.
Oh, the predatory nematodes. Yes, I love working with predatory nematodes because they're so interesting, because there's this other type of nematode out there that feeds on these bad plant parasitic nematodes. However, after quite a few years of research on it, we're finding that it's very challenging to raise them up to a large enough scale where that you would be able to make a commercial product to give, I can't go out there and give you, you're like, here, add these to your soil. That's not been possible at this point yet. And, no one's been able to, achieve that protocol. One suggestion that we did have, and what we found, was that the more organic matter that you have, then the more predatory nematodes you have. So we do encourage, having a healthy soil environment, because what happens with predators is once they feed on your bad one, then their population drops. They run out of food, they start cannibalizing, their own. So if you have a healthy soil environment, you have other type of nematodes like decomposers, fungal feeders, and bacteria feeders. So then they have these other good guys that they can eat in the meantime, till the other guys, bad guys, come back, and then they can jump on them. So it's a war down there, but you got to balance it out, just right. But I do want to look at that again in the future. But right now, I'm getting pulled in.
Previous times I had problem with certain plants, specifically orchids, oncidiums, was very problematic, and somebody I didn't know anything about it, sent me some from the mainland, which I literally had to spray on the plants.
Right.
As beneficial nematodes. Unfortunately, I didn't have a long enough trial period to see any benefits.
Okay, that type of beneficial nematode is the other type that I mentioned. That's insect parasitic nematodes. So right now, there's a restriction on bringing those into Hawaii. So yeah, I've been doing some research. We did some surveys to document their existence here already. So we have, they're quite prevalent, throughout the islands. So we're working on projects like that, to move forward with, hey, can we either have somebody here raise these for the growers, or, can we get some exemptions to be able to bring in these commercial products, because they're commonly used, all over the mainland, and that's a resource that we're not able to use here right now. So that's another aspect of my studies, which I think is very, very interesting.
Roxana, in management of parasitic nematodes, you mentioned starting with clean cinder. Of course, for the Anthurium growers here, we've got gazillion yards of cinder in the beds at the present time. So, you know, as you know, we've been very interested in trying to find ways of cleaning the beds. We've tried the steam, which worked, but it's not very practical. You want to talk about the hot water effort?
Yeah. So I love the hot water, and the growers do too, because if you had a hot water system, that's an affordable piece of equipment that, you know, each grower could buy on their own. However, there's some challenges with the movement of the water and the heat transfer through the cinder. So, you know, that project has kind of taken a back seat right now. I think it has a lot of potential though, and I know there's a lot of interest in doing that, and I do want to pursue that some more as well. One thing I would like to bring up, since you mentioned that though, is we've proven in a number of different field trials that steam sterilization is absolutely the best way to clean your cinder. So you can get 100% eradication, but the equipment is extremely expensive. Now, if this is something that HFNA would like to look into, like with the hot water shower that the university has, that the growers are able to share, if there was some way you could get a grant to get a steam sterilizer, you know, that if the growers could, you know, take turns using, then you would be able to, you know, disinfect your fields maybe on some type of scheduled basis. So that was my most recent thought on that I think there's potential, because the university has done that previously with the hot water shower containers, and allowed the growers, you know, to utilize that when they need it. So, because I know that it can be, you know, very costly to buy that type of equipment. Yes.
So this is a silly question. So many of our growers right now have not been able to use a nematicide like Nemacure for over 5 years now, as you know. So many of them have been top cutting if they're not in a tissue program, and they're reusing, and possibly 2 times already.
Right.
So is there something that you can help our growers with, where we can actually either hose in or spray on, or go with irrigation that could band-aid for now until we get the recommendation, you know, of the final treatment, something that could help us?
Right. Yeah, the only thing that I can say right now, because there are limited options for controlling this pest, is the hot water dips. When you do make the cuttings, get rid of those nematodes, you know, while you can, because even if you can't, you know, this doesn't look like they're in the gobo itself. I've seen some ridiculously high numbers, tens of thousands just in one, you know. So I think if you can, you know, cut your nematode populations down, then you're going to have a less, you know, problems with yield. You'll have less damage. And yeah, and just trying to maintain your plants as healthy as possible, you know, keeping them at the optimum production levels. But it looks like, I think, within the next year that they're trying to put ornamentals on the label for this indemnify. So
hold our breath. It's close.
There. I know we always say that. It's right around the corner. But this is probably the fastest thing right now.
Is the registration with the state of Hawaii, or is this federal?
It's for the label, the product, yeah, the federal.
That takes a little bit longer, I guess.
Okay, well, thank you very much.